Hi, in this video we are going to quickly talk about eukaryotic cell. Let's get on with it. So, if you talk about any eukaryotic cell, we'll divide the following video into three parts. What are three parts? We will first talk about the cell boundaries, and then we'll talk about the cell regions, and especially the nucleus. And then finally, we are going to talk about some of the important organelles that are there in the cell. So, here in the front, you have a cell. Now, each eukaryotic cell has a boundary which is known as the cell membrane. So, this boundary that we just drew is the cell membrane. Now, each cell membrane is made up of a bilayer. It is a bilayer. Why do we call it a bilayer? Because there are two layers, and the layers are made up of phospholipids. That means lipid molecules having phosphate group at their ends. Now, because of the phosphate group that they have at the end, that is why the phosphate makes the both the outer and the innermost surfaces of the plasma membrane or the cell membrane as hydrophilic, water loving, whereas the inner surface hates water. They just don't want water. So they are hydrophobic, right? Now this makes sense, so that not everything and anything that is soluble in water can enter through the cell. And as you know that water, because of its high dielectric constant, right, water has got a very high dielectric constant which makes a lot of things soluble in water, and we don't want everything and anything to come through, right? Now, there also exist in the phospholipid bilayer protein tunnels. So what are these structures? They are protein tunnels. Now, these protein tunnels or proteins can do several jobs. Some of the proteins can form tunnels like this one, and by forming a tunnel, control it can control the entry or exit of substances, or they can also form receptors, provide identity to the cells, right? So this is a cell membrane. Now, in some of the eukaryotic cells, besides a cell membrane, we also have a cell wall. A cell wall is an extra protective layer that grants the cell a protection against the osmolarity changes. So it protects the plant mechanically. So against mechanical pressure. Now this cell wall is made up of different substances in different organisms. For example, it is made up of cellulose in plants and even in algae, whereas it's made up of chitin when we talk about fungi, right? Now, this was the very first thing that we talked about. Remember, we will be talking about things in three-step manner: cell boundaries. Here we talked about the cell membrane and the cell wall. Now, talking about the regions, right? So you can clearly see that there is this region inside. There is this region uh, inside the cell, right? Inside the cell membrane, but outside the nucleus. So what is this? This is nucleus, but the zone between the nucleus and the plasma membrane is known as cytoplasm, and this cytoplasm is made up of several stuff. Now what is that several stuff that makes a cytoplasm? Let us quickly talk about that. Okay, now cytoplasm can be made up of fluid that's called cytosol. Now it can also have cell inclusions, small granules, of starch or protein or other stuff inside the cytosol, and it also has membranous organelles. Organelles are like chambers. Each chamber carry out their own function. We'll talk about some of the organelles later in detail. And finally, all eukaryotic cells have a nucleus. Now, to study more about nucleus, let us go to the depth of it. So, nucleus is always surrounded by a nuclear membrane. Now, nuclear membrane is double walled. Remember, plasma membrane was bilayer, it was having two layers. The nuclear membrane is having two walls, and each wall is a bilayer, right? Each wall is a bilayer. Remember that. Now, what is inside nuclear membrane? The fluid called nucleoplasm. In this nucleoplasm, we have some dense structures which we call as nucleolus. What is the job of nucleolus? The job of nucleolus is to make a lot of rRNA, ribosomal RNA, that makes ribosomes. And we know why are ribosomes important, because ribosomes are protein factories. They make protein. Now, what's inside the further, what's the last component of nucleus is the genetic material that is supposed to carry all our information. Now, that material is a deoxyribonucleotide. Several deoxyribonucleotides join end to end to form an invisible thread-like structure called deoxyribonucleic acid. Now, you won't be able to see this thread when in a normal cell. This will just appear as a blob and nothing else. You won't be able to see this thread, but this thread is not so loosely left out in the nucleus. It is wrapped around proteins in eukaryotes called histones, which happens or which enables to condense the genetic material. And this condensed state, this material is called chromatin, which is DNA plus protein histones, right? So histone proteins plus DNA, they form this. Now, they further condensed to form looped domains. They form loops and super loops, right? So looped domains, and that further condenses while the cell is dividing to form a structure called chromosome. Now, this chromosome is visible, but you can only find it at metaphase of the cell. Now, what is metaphase? Metaphase is a phase of cell division when all the chromosomes are at the center of the cell, right? So when you have chromosomes at the center of the cell, that is when you can see it. But I know your brain must be asking question that uh, always almost in your books you see two arms of the chromosome. Why? Where does this second arm come from? Because we said that chromosomes are only visible at metaphase when the chromosomes are at the center during cell division. And before cell division, you need to make a copy of the DNA because DNA has all the information, right? So that is the whole logic of having that butterfly shaped structure. I hope this makes sense. Now, quickly talk about a few important organelles. So starting from the organelle that almost engorges the whole nucleus, and that organelle is endoplasmic reticulum. Now, endoplasmic reticulum, or simply called an ER, can be of two types. Smooth, as you can see that these ER are very smooth, but some other types of ERs can not be very smooth. Why? Because on their surface they have these dotted structures. What are these dotted structures, which are also found on the nuclear membrane surface? These dotted structures are your ribosomes. And what's the function of ribosome? We just studied, that is protein factories. So these are RER, rough endoplasmic reticulum, which makes a lot of protein, and this is smooth endoplasmic reticulum, which can do a host of different jobs. It can synthesize membranes because it synthesizes a lot of lipids. It can store calcium if you talk about your muscles, right? It can also carry out detoxification of certain drugs, right? Once these proteins are made, they are shipped in small packages. So once these proteins are made by the ER, they are shipped in small packages. So these are small packages inside of which lies the protein. Now these proteins are modified. So they are modified by yet another organelle chambers called Golgi. Now Golgi packages some of these proteins as lysosomes. So they may package some of these proteins as lysosome. What is the job of the lysosome? That is to digest unnecessary part, right? Digest unnecessary part of the cell or something that has lost its function. If too many lysosomes burst in the cell, even the cell can be digested as well. That is why you call it a suicidal bag. Now, there are other structures called centrioles. Now, centrioles in a centrosome, they have a role in cell division, and they also have a role in organizing the cytoskeleton of the cell. Now, what is cytoskeleton of the cell? It is an array of protein networks that give the cells a shape. Now, let's talk about the organelle that gives you a lot of energy, and that organelle, yes, that's very correct, is our own mitochondria. Now, mitochondria has an outer membrane, a membrane on the outside, which is very fairly smooth. Then they have an inner membrane. Now, you can clearly see that the inner membrane is folded inwards to form shapes called cristae. Why we have more foldings on the inner membrane? Because we have structures called ATP synthase. What is the job of ATP synthase? The name itself is telling you it will synthesize a lot of ATPs, right? Now, the inside of the inner membrane, you have the matrix where a lot of Krebs cycle enzymes are there. What is Krebs cycle? That involves breaking of pyruvate, a product of glucose breakdown. Now, between two membrane, you have the intermembrane space. Now, on a similar lines, we must talk about another organelle that is very unique and found only in plants, and that is chloroplast. Again, for chloroplast as well, you have an outer membrane, a membrane on the outside, and an inner membrane, a membrane on the inside. Now, apart from them, you have a third membrane system, bags called thylakoids. Now, thylakoids are embedded with molecules of chlorophyll that are going to absorb light and create chemical energy that is going to be used to synthesize carbohydrates. But thylakoids are found in stack. Now, what are these stack called? Granum, singular. Now, obviously, you can see there's not one granum. There are many granums. No, so you don't call them granums. You call them grana, plural. Now, outside the grana and inside the inner membrane, you have this space, right? What should this space be named? This space is called as stroma, and through stroma there are connections in between different grana, that's called fret. They are also known by the name stroma lamella because they are connections, right? So this organelle is chiefly found in plants and other algae. Now there's another organelle, a vacuole. Now, vacuole can do different jobs in different cells. Now, if you talk about amoeba, an amoeba has a contractile vacuole. This contractile vacuole can pump out the extra water that you have. Now, in case of plants, these vacuoles can store a lot of other substances. So, there's a lot of storage going on in some plants. Uh, the storage can be of food, the storage can be of pigment, it can be of waste products as well. Plants, though, always have large but few vacuoles in plants, right? On the contrary, animals, on the contrary, if you see, animals always have many but smaller vacuoles. So I hope this establishes some clarity between the various organelles of the cell. That was the third part of this video. I hope this makes sense.
Thank you for watching this video. Don't stop now. Check out the next biology topic. More is coming. Subscribe and stay tuned.